when I was asked to do this, I said yes, because in a lot of ways, this meeting or the variants thereof are where I came from. And then I started thinking about what I was going to talk about and realized I don't actually do that anymore in exactly the same way. However, I think I'm going to be getting back to it. I'm going to try to prove that to you. Because I've been trying to figure out how to make the engineering of new function in cells more reliable. And the bugaboo, anyone who's a naysayer for the field of synthetic biology, which aims to do that, basically says you can't do that because of that all those connected, Bush diagrams everyone here likes to show. I try to put something in there and everything's affected. And the question is can we break that down? Can we figure out what the pieces are that make things more or less reliable in cells? And I'm going to try to argue that we're going to do that. And I'm also going to argue that it's important that we do it because society wants us to. So I find myself having gone through many careers, and I'm now an engineer of a sort. And I was thinking about what it means to be an engineer as distinct from a scientist. They're not that distinct. But really, engineers are scientists of effect. That is, they want to create an effect. Now, you can call people who make a drug that cure a disease engineers of a sort, I suppose, in that sense. But our idea is to create an effect. But engineers do so in a way that from the outset, gains trust. And that trust is very important, because it's not just the trust of the people we're affecting, which is what a drug has to do. It's the trust of the other people in the field. When I say that something works in way X, it should work in way X. All of you who are algorithm designers know this. When you write your algorithm, it better work as well on on someone else's machine as it works on yours. Your machines that you build should work the same. And so it's these two things that we have to do. Most cellular engineering, most genetic engineering for the past 40 years has focused on effect and not on trust. It has been, I'm going to do whatever I can, in whatever way I can, to get the effect that I want. I'll breed, I'll select, I'll make libraries and screen. I will do anything I can to get the effect that I want. Same with drug design in a lot of ways. It's gotten better, but it's, whatever it takes to get the effect I want, I will do it, without having to think rationally about what the trust and effect would be. But I figured that is what I want to do. And back when I was a youngin' and talking more at sort of systems biology and network biology conferences, I spent a lot of time talking about what it meant to understand something. And to understand, in those, at the time I said it meant the ability to predict what it will do, to control what it will do, and design in the medium. And design is what I'm going to talk about today. So we're going to start with why I think it's important and why guys like me should be thinking about this. And I'll talk about sort of designs, futuristic designs in synthetic biology. So when I started my lab, I was fully computational. And now I'm probably 70, 80% experimental, which is a big change for me. And what I can do now is pretty extraordinary in terms of what I can do to make an entire chromosome or an entire genome of an organism. And the question is what can I do with it? And I remember back in the day, around, I don't know, 5 years ago, 6 years ago, maybe more than that now, we proposed a therapy for HIV that was basically engineering a viral chromosome to be a parasite for HIV. And we could build it now. I can't promise it won't cause any harm, but I can build it. Right? So we need to get trust in line because we now have the technology to do things very quickly that could have profound impacts on people. I'm going to show you one which is a bit of safer design that we've made some progress on that's kind of interesting. And we talk about really smart drugs. I mean, not just drugs that are decorated in such a way that they recognize the right antigen on a cell or they target a particular enzyme, but actually reason, as much as the bacterium can reason, about its environment, and only when conditions are exactly so, implement the kill for the cancer cell that we want it to kill. And the idea is that bacteria are better than, for example, smart liposomes because, well, smart liposomes can do things like if antigen, then fuse. They can't do if this antigen and not that one, but this one and not that one, then do it. So bacteria can. They can decide to invade or not a cancer cell. They can decide once inside to make sure. And then once that's done, they can synthesize the chemical at site and do the job. So they're really, really smart organisms, so to speak. When we sat down to do this, and this was myself and Chris Voigt and Chris Anderson back when we started in my lab, and Chris was just leaving to go to UCSF, Chris Voigt was, we said, well, we're going to break this down into a series of problems. We're going to imagine that we're going to inject the organism into the bloodstream, and it's going to circulate around. It's going to find the tumor. It's going to grow in the necrotic region, and then it's going to invade and do its job. And so we broke it up into a series of block diagrams. That we would have to build. We'd have to be able to sense that we're in the blood. We'd have to make sure that when we're in the blood that we, weren't causing inflammation and sepsis, and we weren't getting eaten, and we could circulate, and we weren't growing. That would be bad. We had to know that when we're in the tumor microenvironment, that maybe we could grow, but perhaps we should be able to sense we're there and then turn on adhesion molecules and begin to invade. And then we should be inside the cell. We should be able to figure out, hey, we're inside a vacuole, because probably we're going to get in by being engulfed. We're not going to poke through the membrane. We're going to be engulfed by endocytosis. We had to break through the vacuolar escape. We had to implement a kill module and then send them out. We had to make sure that it was safe. So, for example, it would be pretty bad to make a tumor-killing bacteria that spread from person to person for a number of reasons. So you wanted to make sure it was linked to that human being and so on. So how would you do it? Basically down here are a set of actuators we had to find. Up here are a set of controllers that logic, that if the right logic is met, you implement these systems. And up here are the sensors, the things we have to sense. Talk about a few of them. Just to give you some sense of what actuators look like. So when I say this, it's concrete and not airy-fairy. This is an example of just a growth controller where we have a gene that when you express it, it can scavenge iron even from hemin. So it can grab irons out of these high-affinity binding proteins. And because of that, the cell can grow in blood. So we prevent it from doing so by turning off that gene when it's in blood and then turning it back on when it's in the necrotic region. So you can use it. So in this particular case, it's very simple. You have this gene called TON-B, which is a cofactor for iron transport, more or less high-affinity iron transport. And we can show that the cells, when you expose them to no iron, can't grow in wild type or with this deletion. And they can grow in wild type with hemin, but not when TON-B is deleted. And that's one of our actuators. And so on. We have ones for linking it to human being. Here it can't make a very critical component of its cell wall. It's a metabolite that we don't make. It's a metabolite you don't find free in the world. And so we have to feed you this metabolite so it can survive. A safety switch, so to speak. We have a capsule. This is actually not working as well as we'd hoped. But when you look at E. coli in the laboratory, it looks like that. When you take E. coli from a human being, it looks like that. Within a human being, it has to deploy a capsule. All the O and Ks you hear are compounds that are in capsule and lipid membrane and polysaccharide. And they allow the cell to survive, more or less. They prevent it from being endocytosed by neutrophils and macrophage. It prevents it from causing inflammation, and it allows it to grow. Unfortunately, it prevents it from doing other things, like adhering to cells in a lot of ways, at least by our mechanisms. And what we're showing here is that we can show that if you dump a bunch of blood on these guys, they will survive if they have the capsule, and they will not survive if they don't. So just a capsule actuator. And then we have this invasion actuator, and this is something discovered by Stanley Falco, and it's one of these great proteins which you can put it on a styrofoam bead, and if this protein is on the styrofoam bead will invade cancer cells because it's a strong beta-1 integrin agonist. So it binds to beta-1 integrin and gets sucked in, and it's pretty cool. So if you can make the bacteria express this on its surface, it will bind to beta-1 integrin, and off it will go. And luckily, you can tune this because there's all these cancer cells which have really abnormally high levels of beta-1 integrin. And so you go, well, great, it's a great marker, let's use it. And so all these graphs here are different cancer cell lines and then the normal cell line, where in all cases of the cancer, we can invade when we have invasin. All the white bars are with invasin, black bars are without invasin. And then the normal cell line where it can't do it either way. So again, it's a actuator for invading cancer cells who have the biomarker, lots of beta-1 integrin. So now we have these different actuators. Let's go up a level and talk about logic for a minute, just to give you some sense of what that looks like. The logic here, of course, is specificity. All of this is about specificity. You want to get it to only do these things when the conditions are met. And so this is a two-dimensional specificity diagram, obviously really conceptual. But the idea here is that in tumor, glucose tends to be low and oxygen tends to be low. Actually, it's probably not true. It's probably glucose is high and oxygen is low. But so the idea is that if you want to only do your thing in tumor, you'd have to be down in this quadrant somehow, and then you turn on invasion. That would be the idea. Obviously, this is multivariate. There's many inputs, and you want to use all of them. How do you find those? How do you find the environmental sensors for those topics? Well, you guys know as well as I do. You use those big network bush diagrams, and you find the ones that are important for the conditions that you want. Very simply, it's just, do gene expression, do something that shows which genes turn on under these conditions. In this case, it was really dumb. This was an oxygen condition. We know that at low oxygen, which occurs in the necrotic region, this gene turns on 20-fold, and it's pretty much off otherwise. We had very quantitative data on it. This is a spotted microarray for some reason, but you have very quantitative data on this. And it's pretty good. So we said, OK, fine. And we have other things. It turns out this is a great story. If you take any bacterium, give me a bacterium, and you inject it into the tail vein of a mouse. Unfortunately, it doesn't work the same for a human being, but for a mouse, it'll be all over that mouse in about 20 minutes. It's all over it. And in the space of a day or so, it will have localized to a tumor. In this case, it's a tumor on the leg of the mouse. And it's actually about 10 to the third higher in density in that tumor than it is anywhere else. So it's cool. We can use density sensing then, right, as a sensor. So in this case, we borrow a quorum sensing system, not the one from Vibrio fischeri because it's oxygen sensitive, and we're trying to do this with low oxygen. But there you are. And the idea is that as you increase cell density, you increase the amount of activity from this promoter. And in this case, this gives you an example of where we started doing ad hoc engineering. We wanted this switch to turn on at a density that it was not evolved to turn on at. We had to figure out a way of making it turn on at a given density. So we didn't know how to do that, and so we made a library of gene expression elements and then hoped to find one that worked. So we screened a bunch of them. This is a saturation region on an RBS start site. And sooner or later, we found one that pretty much would only turn on when cells were at a density that you'd find in a mouse tumor, bluntly. And so we can turn them on. And here's the percent invasion as a function of density. And so now we have a very simple sensor actuator system where at a critical density, then you invade. Now I'm going to make things more complicated. Turns out I can do the same thing I could build the same sort of sensors for anaerobic growth, for arabinose, which is very high in the tumor necrotic region, and so on. And now I want to build an AND gate. We can say when you're at high density and oxygen is low and arabinose is high, then invade, because that's pretty specific for being in the tumor necrotic region. So how do you build these sorts of gates? There's many ways. This is a very old one. We have many, many more of these now. And Chris Voigt just published a wonderful paper on a 5-input AND gate. But this is an example so you have some sense of how they work. And I'll get to the things that are interesting to you guys in a minute. So you have two promoters, for example, two of these things I just told you about. They feed in to drive these two genes. In this case, it's a tRNA suppressor, an AMBER suppressor, and a T7 polymerase gene that has two different stop codons in it. And only if the suppressor tRNA is expressed can you get past these stop codons and make a functional T7 RNA polymerase, which then makes GFP. It's a really dumb AND gate. Works really well, but it's a really dumb one. Wouldn't use it in a normal situation. The point is you can sort of see here what happens. Plus SAL, you get nothing. Plus ARA, you get nothing. Plus them both, it turns on, and it has a sort of transfer function, and we can predict it and all that good stuff. So now we're going to put it all together. I'm going to show you how you build a more sophisticated system. And I want to give you some sense of why of this, because it's after this that the problem of context becomes really important. I've just shown you that a bunch of ad hoc discovery and a bunch of ad hoc engineering to get everything in to the right levels and thresholds and things. And now we're going to do a bunch of ad hoc engineering of a very large piece of DNA to try to get this thing to do something very clever, which is to generate a payload, to invade a tumor cell, to detect it's in a vacuole, to break open that vacuole, and then to deliver the system both to the cytosol and to the nucleus. I'm going to do all those things simultaneously, one after the other in order with nothing going wrong. Okay. So we have these different devices. We like to call them devices. They're just little gene expression cassettes. So this is a lot. This is 140 kilobases of stuff we're talking about here. So obviously, we have this invasion device. I told you about it. It's pretty easy just to deploy it at the right time through these AND gates. We have to do this vacuole sensing device, then the self-lysis device, where the cell kills itself, and in doing so kills the vacuole. Because we're going to release ourselves and then release the vacuole. And so each of these things has a whole bunch of genes associated with it that I won't go through in great detail because it would take forever to describe it. But we can do these sort of unit tests. So the idea is, what you're going to see here is full and magnified views of a single cell, When you see things magnified, we're looking at a cluster of vesicles in that cell. And the only thing that's glowing is the bacterium. Okay? So that's what you're seeing. And so what you're seeing here is 4 different experiments where we have a payload device, which in this case is just 2 fluorescent proteins, GFP and RFP. We have the invasion device. And so if you have the first device, all you should see is a bunch of bacteria ending up in vesicles. And lo and behold, you see all these little point stops, and there's a bunch of bacteria stuck in a vesicle. Kind of boring. They're all stuck together in this little ball. And they're nice little rod shapes, meaning nothing's happened to them. Here's B, where we have now a vesicle sensing device and a self-lysis device, and we thought that was kind of cool. And what happened was we just circle this little area, and we find there's the vesicle, and there's a bunch of bacteria, but they look kind of not self-lysed. They look kind of bloated. And that was very sad, because in vitro, as outside the cell, they lyse just really, really well. But inside the cell, not so much. Inside the vacuole, not so much. So we mucked around, we found another gene, and lo and behold, when you put that gene in, suddenly the vesicles are completely filled with GFP, little balls of GFP and RFP. So we've succeeded in lysing our cells. Bonus. Finally, we had the vacuolysis device. That didn't cause any problem at all. And what's really nice about that is when we put it all together, in the end, we could deliver GFP to the nucleus and RFP to the cytosol, and the whole process went as planned after a lot of mucking about. Just lots of tuning and mucking and a lot of work. So that's very sad that it took this long to do this. It took a lot of time and a lot of effort, and there it is. So I want to make this much, much easier. I want to make it one thing after the other. And this requires knowing 3 types of things, and I'll talk about in a minute. So these systems almost work. They require this massive tweaking, many years of development. They cannot be selected. So you can't do these things like, I'm going to select for more production of X, because I'm going to select for a bacterium that kills tumors. Anyone want to volunteer to be in that screening population? It isn't going to work, right? You can't do it. It's too slow, and it's dangerous, and it's dumb. So you can't do it. You've got to get much more rational about these things. And the reason why is that the individual elements are very context dependent. They depend on where they are, and we need to discover what those pieces are. So there's 3 pieces of context we have to discuss. There's the immediate context. So if I have a promoter and I put it into a new piece of DNA, it may function like it functions in that piece of DNA, but it may not. And I want to know what causes that to happen. There's host function. As you know, any process that we have will rely on host functions, if only because it needs to make the proteins or whatever to do the thing. And that means you're coupled to amino acids, and you're coupled to RNA, and you're coupled to all this other stuff that the host has to make for you. So you're coupled to the genetic background of the host, codon usage, growth, things like that. And of course, the environment. Obviously, you guys all know this. Sometimes I have to tell this to my engineering colleagues, but I do want to bring it home. This is just gene expression correlation over a number of different environments. And you can see they are very different states. We all know that. But they're very finite. And so we can kind of get a sense of what they are. We can kind of map all the sigma factors and how they're changing all the promoters. And we can say, well, these sigma factors are indicative of this state, and these sigma factors are indicative of that state, and so on. And we can begin to make a map of all the logic the cell is using to deal with these different states. So we're starting to make an environment-to-host map. And we can do this at extremely high throughput now. We're at the point now where we can introduce transposons at between 4 and 20 base pair resolution across an entire genome within the space of a week. We can then run 300 culture conditions or more, or a bunch of different conditions, and we can then screen them by sequencing and score whether or not the loss of that gene improves a phenotype or depresses a phenotype, depending on what phenotype we're screening for. And so if it's yellow, it improves the phenotype, and if it's blue, it depresses the phenotype in some way. In this case, it's almost all growth. And we can use this to annotate genomes and do a lot of cool stuff. But we're getting to the point now where we have enough of this data in enough conditions that we can even do things like you guys are familiar with. So this is an equation all of you people who've been working on network inference will be aware of. Basically, you have the state of some gene at some index and condition or time or whatever is dependent on its whatever it was last time step plus some nonlinear function of interaction with all the other genes in the system. Okay, that's what it is, plus some noise, plus other stuff. And you have this sort of function, this generic function of regulation you put in there. And basically what we do is we fit models like this to across these conditions where in each condition we can ask whether or not the strain missing this gene did better or worse. And we can score that by knocking out the coefficients about that gene because it's no longer present. And it turns out you can begin to constrain these networks. Now, I'm not going to go through this because it's not the point of this talk. It's thought you guys should see it, that it exists, which is that, here's an empirical regulatory network in Schuanella, and here's the inferred linear part of that network. And we don't do terribly, we don't do wonderfully, but people haven't inferred things from fitness before, so we're pretty happy this is the first time that this has happened. Work by Xiaodong Wang at Columbia with Michael Samoloff in my lab. So we're beginning to get these maps that map environment to host factors, and then host factors to each other in those environments. So context-dependent host factors, and we're making these formal models that make this work. We now have to ask how an implanted function is in fact affected by those host genes. So we have environment to host gene, now host gene to our implanted function. So our implanted function is a very simple function for this time. It's just three different fluorescent proteins, two of them driven by the same promoter and 5' UTR, one of them driven by a different one. So you're controlling for coding sequence effects, and you're controlling for promoter effects and things like this. And then what I did is I passed this into this deletion collection, in this case an E. coli deletion collection, and then asked how each, well, actually this is not that. This is passing that same system into 8 different cloning strains of E. coli. They're all the same species of E. coli. One's a B strain, the others are K strains. And then they're all just different variants of each other. And what you're seeing here is the YFP versus RFP expression from that probe construct. And the point is that they vary extremely widely over the different strains of E. coli. Anyone who's done a lot of synthetic biology knows this is the problem. So there's a lot of host background dependence here. They're otherwise just the same. And second of all, you can see that there's spread in the YFP, GFP, the RFP, YFP background. And in some cases, it can be very extreme. In this case, it isn't. I'll show you one where it is. Here's where we put it into the deletion collection. So you put it in deletion collection, and each one of these is a cherry versus Venus fluorescence under a different gene deletion. And what you're seeing here, for example, is that NAR-L makes things overexpress, and you knock out key Y and things underexpress for some reason that I don't understand. And there's a whole bunch of guys who, if you knock them out, express cherry, then they express Venus, and so on. So there's all this host factor dependent expression, and we're beginning to be able to put those together into networks that explain how the host impacts your implanted circuit function, in particular all the expression functions. Now, that's all about a probe on a single plasmid. We can do the probes on a vector that inserts into the genome. In this case, I just want to give you a sense of one of the systems we work on, because we're doing a lot of medicine today, I guess. So I'll talk about a medical one. This is HIV. HIV can insert randomly into the chromosome, or quasi-randomly into the chromosome. And then you can measure, for example, its expression. In this case, it's a probe with a GFP attached, as usual. And the point here is that when we make this random collection, and you collect clones, and you ask how they work, you find guys who are dim, guys who are bright, and guys who are middling. And you could expect that. And you can build models of this, and we do, to show that you can ask how each feature of the surrounding chromatin affects these different parameters, burst size and frequency and things like this. And you can fit those models, and they work pretty good. And in the end, so this tells you what varies under various conditions. They're extremely important. These different small features of the surrounding chromatin affect very strongly this burst size and burst frequency variation. They affect it very, very much, including the mean. And we can take these 3 variables, mean, burst size, and frequency, and plot them in such a way that we can say that, well, guys who have those parameters never turn on. Guys who have these parameters out here just turn on and make lots of virus. And guys in there stay off long enough to be latent, to be captured in the memory T cell state, but can also come back on again when reactivated. And so it's parameters in this region that we think underlie a lot of latency. Okay, so now we want to get rid of all this natural variation. I've just told you we have all these things, the host and the environment and the chromatin environment, which cause all this variation, and we want to get rid of it because we can't possibly learn all the possible variations that can exist. Can't do it. So we have to eliminate them as elements of variation. I'm going to show you how we're going to try to quantify them and then fix them. So we start with this very simple genetic context, and what we call the expression operating unit, which has these different pieces to it, promoter, 5' UTR, CDS, for example. If we take the same promoter and the same UTR and put GFP or RFP on it, we get a point here. So this GFP and this RFP are expressed at 56 and 33 units respectively when expressed from the same promoter and UTR and everything else on this thing. And then this is another pair of promoter and UTR, and this is another pair. And if you look, theoretically, you'd want to be able to say if a strong promoter and a strong UTR, you have a strong expression. But that's not exactly true. We have about a 0.59 correlation, which is not bad, but not perfect. So let's talk about this immediate context. This is all work that we did with the BioFab, which is an experiment in open source biology. And I want to get a set of parts that are complete, which I can make any function, in this case gene expression function. It should be scalable. I can make complex circuits. It should be homogeneous. I should be able to measure a member of this family once, and it applies to all the rest of the family. And I want invariance. It holds in different contexts. So let's talk about this. Ideally, we'd have all these different parts that we've characterized, and when we put them together, they behave as predicted by the individual models for the individual parts. That's the idea. So we, of course, can make these things. We can characterize these promoters in a given context of these two things, and we can get a variation in promoter strength, a variation in RBS strength, and a variation in termination strength, and things like this. We have lots of these parts. Problem is the interactions between them, just genetic context. When you connect them together, what happens? So if I take this RBS and that promoter, what happens? And ideally, what you do is you take a very strong promoter, take a very strong RBS, you get a very strong output. That's what you want to see. And so you should see this sort of decreasing gradient as you get weaker and weaker and weaker, ideally. Non-ideally, you get things that look like this. This is promoters and UTRs, and this is GFP. And yeah, there's sort of a gradient. That's good, but it's kind of noisy. With RFP, it's worse. So two things to take away from that. One, it's not such a gradient anymore. And two, it's very different for different genes. That's a problem. So you can quantify this, and you just do it by ANOVA, nothing fancy. You ask, how much does each element confer the, explain the variation in the output. And you can get models like this, which say, look, if you look at fluorescence, which is the amount of protein more or less, it's mostly explained by promoter identity and by UTR identity, but there's this interaction term between the UTR and the CDS that seems to be a problem. mRNA abundance, you find it's mostly UTR with some promoter. That's a little bit surprising, right? The promoter should make a lot of the transcript, but of course the UTR determines degradation rate, right? So that's very important. Also, you have an interaction term here. And translational efficiency. Once you control for the mRNA abundance, how much protein do you get? There's a very big surprise. It's the promoter, not the UTR. Marcus goes, what? And the reason why is because what people call promoters are not promoters. What people call promoters have lots of other stuff associated with them, including UTR. And those first few base pairs after you start matter a lot. So you can also run it the other way. You can take each promoter and score it. So yeah, they have a mean score. There's stronger promoters and weaker promoters and stronger RBSs and weaker RBSs. And these other bars you're seeing here are models that tell you that, yeah, look, here's 3 UTRs with the exact same main score, the exact same strength. But this guy maintains that same score over all combinations with CDSs and promoters, whereas this one doesn't. We don't know why, but it doesn't. Now, it turns out that we can use this to decrease the number of things you have to characterize to make these models. This is all combinatorial. But at least we can begin to understand that there's some computational dependence here, and that what's more, it's very related to something to do with this UTR-CDS interaction. Maybe we can fix that. So let's take away, ideally what you'd have is you'd have an RBS, you'd vary the RBS, and something that was strong for gene 1 would be strong for gene 2. So if you have a bunch of genes down here, and you had and each one had, you could try an RBS in front of each of those genes, the relative position of that RBS relative to other RBSs should be the same. So ideally, there'd be straight lines across this diagram. That is, if you had this gene with this black RBS, and this gene with this black RBS, and this gene with this black RBS, it'd be a straight line, right? Ideally. This is what it looks like in reality. It's just a bloody mess. If, however, you use a trick that was discovered many years ago, and you use a leader sequence, a little peptide leader sequence in front of this thing, and you embed an RBS in that peptide coding region, then when this ribosome is translating this leader sequence, it breaks up the structure along this edge here. It removes any other factors that are on that piece of RNA, and it reinitiates at this R2 in proportion to its binding constant to that Stein-Delgado sequence, and you end up with a much flatter group of things. So to give you some sense, going back, remember this is now a much larger set of data, but This is without the bisystronic UTR with lots of genes and lots of different UTRs, and it looks like a bloody mess. It kind of has a fade down to the bottom. Yep. And here's a case where we put the bisystronic design in, and suddenly you get a very nice behavior, top to bottom. And to give you some sense of things, if you do that same ANOVA, you find that the interaction between the Shine-Dalgarno and or the 5' UTR and the gene of interest without that bisystronic design has a 16% explanation for the variance. Whereas with the bisystronic design, you've eliminated it. It's less than 1%. So you can begin to get rid of those pieces. Now, it turns out that we can do this. This is when you take now all the promoters and UTRs, and now we get the perfect fades down. So now we can get a 90. So we're getting an R squared of 90 of predicted versus observed of 0.9. So we're hitting our mark very, very accurately now in this situation. Now, it turns out that we can add a whole bunch of precision control elements to this 5' UTR. I don't have time to go through them, but we have all these different regulators that we can put in there that can create logics. And so, for example, if this regulator, when you had an input to it, turned on this gene, and if this regulator, it turned off the gene, if you put them together, there should be some sort of bandpass, for example. But you can find configurations that make ands and ors and nots and nans and things like that. And the idea is to build reliable combinatorial logic at that promoter. So can we form this basis set? Well, we have a couple of these things here. So we have a translational controller, which is based on a small RNA that binds to the Shine-Dalgarno sequence, more or less, opens it up and causes it to be sequestered and degrade the transcript. So it's an off. We have a translational controller, which uses a different mechanism, but also a small RNA control. And the idea is, can we build orthogonal versions of these things that you can compose on the same strand, and things that you can chain together. So it turns out that for both these guys, I can make fairly large families of orthogonal versions by just RNA engineering. And that's a lot of interesting stuff about molecular context. All the people who do mutual information among residues, we use a lot of that to separate out these guys so you get informationally distinct specificity between these guys. We can attach things to the small RNAs that mediate whether or not they have the right structure or not. So we can use the same sort of computational models to link RNA aptamers to those small RNAs and turn them on and off with small molecules or proteins. And so here we do it with theophylline, and we do it with MS2 coat protein and other things. This also is published, and you can look it up. But the idea is that we now have a system where I can turn transcription and translation on and off, and I can do so with different molecules. And I can do it with these leader sequences I just told you about. And so in this case, we're going to do it with a trip leader or a TNA leader. So those of you who are biologists know very well this attenuator that converts the motion of a ribosome on an upstream peptide coding sequence to changes in transcription downstream. So it's a translation to transcription converter. It's all done by pausing when you have a tRNA in there. And we're going to now control pausing with not trip, but unnatural amino acids. So we're going to make tRNAs that recognize unnatural amino acids. And we can do things like make an off switch and make an on switch with these things. We can then characterize them and then predict what the bandpass will look like, and there it is. It all works out just fine. Now there are some, can we make any function? The answer is I think we can get there. So we're at a point now where I think we have a bunch of regulators we can put into a line. We can have this bisystronic design that controls the interaction there. We have a bunch of ways of making this very, very formal and very precise. Okay. So that said, I've shown you 3 things. There's actually a lot more to this that I wish I could show you. We've really formalized this to the point where I can get 4 of these regulators in a row and get a 4-input NOR gate, and I can do all sorts of crazy things with this now. But I've shown you that there's a few things. We've begun to really unravel genetic context. We kind of know how to change, how to control for genetic variation around the elements that we've built and eliminate that from the table. So now we don't have to worry about that anymore. We're beginning to map host function to genetic context, how a host function affects that gene expression. And we've begun to discover ways of orthogonalizing ourselves from these host factors that affect us. It tends to be through ribosome availability and through cell growth, 2 things that it turns out we can buffer against in our gene circuit designs. And we're beginning to map host gene function to the environment. And so now we can understand what the critical host functions are that impact our our circuit function in a given environment and operate against those. If we can begin to use some of the network models that Marcus has put together, for example, or that some of the statisticians have put together to drive that design, we're going to make a huge impact. I want to give you some sense of the data scale here. I have maps like this now for about 10 organisms. This year, working with the JGI, we're going to have probably 50, and next year, 250. For gene expression, we have it for hundreds now of these types of things. For these sorts of things, we're getting to the point now where we can do this very, very rapidly. And so we're going to have very large data sets about this stuff, and we still don't have the right algorithms for really inferring how we should design these things to eliminate these context effects. So help. That's it. I want to thank people who did this work. All the biofab work was really led by Vivek Vitalik and Guillaume Cambrai, and João Guimarães as our analyst. Drew Endy was my partner in crime with that. The RNA elements were Julius Lux, Vivek Vitalik, and Stanley Key, and Cheng Liu. Working with Jennifer Doudna for some of the stuff I didn't show you, unfortunately, and Charles Janowski. Tumor-killing bacteria, as I said, it was Chris Anderson and Chris Voigt, finished up by Jin He. And that very quick thing on the inference from fitness with Michael Samoloff and Xiaodong Wang. Thank you.
Pretty amazing. We have time for a few questions.
So bacteria evolve very rapidly, and so one of the major problems with bioengineering is that you engineer something that works perfectly fine, release it into an environment, and then these things evolve very rapidly not to do that thing anymore, or to do something else. So what kind of safety mechanisms can you build into your bacteria in order to essentially keep a certain fitness function constant?
Fabulous.
And then the second question, the second question I had is that, so obviously, bacteria are very complicated, so have more chances, in fact, of introducing different mechanisms of regulation, and you have to control more things. So why not using viruses to do exactly the same thing? Essentially use viruses that can introduce cassettes that can control, for instance, release an oncolytic protein as a response, for instance, to some number of sensors.
Fine. Let me answer the second question first because it's easier. So people are, of course, do have oncolytic viruses that they use for designing these things, and there actually was a very famous movie you may have all seen, Legend, in which one of these oncolytic viruses caused people to become zombies, which would never happen in reality. But it was based on a real research program of using a measles virus to do this. Biggest problem with viruses is there's two problems with viruses. One is, of course, that they're a small payload. They don't do a lot of logic. They're basically affinity agents that can insert and do things. So basically, it would be a very simple piece of logic, and it would get things done. But there's some hope for that. Ron Weiss has published recently a beautiful RNAi-based logic circuit that distinguishes between different cancer cell lines. And that is all of a size that could be loaded onto a virus and made. But how do viruses express themselves, and how do they get there? And the answer is that you can't do very well. First of all, you can't tell. It's very hard to program which cells they enter, other than the ones they're naturally designed for. And then second is that they tend to either insert randomly or have variable expression levels on their entry, and it's hard to build circuitry to make them do their job. Third, they're hard to get rid of after they're there. And fourth, they tend to mutate if they propagate, if they're not propagating. Right? So bacteria are better that way. Now, fail-safes for bacteria. Typically, people do things like safety. A lot of safety is about containment, and I won't talk about that because that's kind of obvious. You have a bunch of ways of killing the cell, and you hope for the best. Right? Mutation of the circuit and maintenance of fitness is extremely important. And there's two approaches people are beginning to take. One is to build circuits that are themselves rationally adaptive. Think of them as cellular neural networks. And there's designs out there for making this happen where it learns based on sensors of its own physiology what's going on, and it readjusts expression based on that. I think that's kind of cool. The other one is adaptive mutation, but very focused adaptive mutation. You may have seen a wonderful paper from Eric Klavin's group by Rob Ebert, where they figured out how to use slip coding in between the RBS and the gene, so that when the DNA polymerase comes over this region, it may skip over an AT or two, and therefore cause different lengths between the RBS and the gene, mediating its gene expression levels. And so you have these adaptive tools. Now, I think there's a third one. This is floating around. There are certain proteins that are guided to DNA by RNA strands, a bunch of them. right? And so you can imagine all these RNA strands floating about that are surveilling for the integrity of the implanted circuit. And if there's a mismatch, these proteins come in and cleave the genome up, right? And so people are thinking of things like that. So I think there's a lot to be done there. I think usually it's not unnecessary, but I think it's a really good active area for discussion.
Great.
Yeah.
Any other question? I have a quick one.
Yeah.
So you had these beautiful pictures of the bacteria basically lysing, right?
Yeah.
Right at the right time, payload. Have you tried going? Did those go through the tail vein, or have you tried going through? No,
Yeah,
right?
Yeah. No. Well. So remember, I made this very quick comment that the capsule didn't work.
Oh, right, right.
So
okay.
we put them in the damn mouse, and they get eaten like that. And that's really sad because mice have wimpy immune systems.
Mm-hmm.
So we sort of backed off as we try to understand how capsule function works, because that's
Yeah. It is interesting because moving from lab to like in vivo
to an animal.
When you put up the capsule, I thought, wow, yeah, that's something we need to. Yeah.
The problem is that the capsule isn't done yet.
Mm-hmm.
People think it's done. It isn't done. There's deeply missing components, and they're important.
Okay.
Yep.
Perfect. Thanks so much. Oh wait.
Thank you. One more?
One more, real quick.
Sure.
Sure.
I'll try to make it quick. So the power that you have in the control with bacteria delivering payloads seems really compelling. And but I was just curious. I'm sure you've thought about applying this to different things beyond and besides cancer, even. And so, for instance, quorum sensing. How would you do that if you're not targeting a tumor, but maybe just certain types of cells or certain tissues?
Yeah.
Are there other types of specificity that you can use, or what are the other challenges?
Yeah. Well, so there's lots of so obviously there's a few really tempting areas. Gut microbiome is a really good one. People are beginning to map where microbes are and what they are in the gut and how much they impact your health, which is a lot. And so you can imagine engineering organisms that need to sense that they're in the gut and do things. So there's lots of each tissue has a lot of chemical and physical markers that differentiate it from other tissue. I think that the things that we're missing. I think there's lots that we have chemical sensors for nearly anything. We have lots of sensors for for quorum and density and other bugs. I tell you what we don't have. We don't have sensors for adhesion. We don't know how to sense that. We don't have sensors for stress in a bacterium. In floppy cells we do, but not in the stiff cells that a bacterium is. If someone could give me something that turned on a signal inside a bacterium when it adhered to something, it would really change the way that we could do that type of engineering for tissue-specific engineering. Because then, if it bound to this and that simultaneously, and it was bound, really bound, it would be very specific to the thing it was bound to. And that would be very important. So, give me that.
Thanks again.
Yep. Oh, you want those microphones.